the revolution of genetic engineering, and the promise of gene editing. The history of science is marked by discoveries that have fundamentally transformed our understanding of the world and our place in it. In the 21st century, genetic engineering and, in particular, CRISPR gene editing technology are emerging as tools of unprecedented power, capable of directly intervening in the fundamental code of life, DNA. The possibility of editing the genetic material of living beings, from microorganisms and plants to animals and humans, opens up a vast range of opportunities. Imagine the ability to cure genetic diseases at their root, not just alleviating symptoms but correcting the underlying genetic flaw. Visualize the creation of agricultural crops more resistant to pests, droughts, and diseases, increasing global food security. Consider the potential to develop new therapies for cancer, modifying cells to attack tumors more effectively. And on the more controversial horizon, envision the possibility of modifying hereditary characteristics in human embryos, raising profound ethical debates about the limits of human intervention in nature and the future of our species. This script will delve into the fascinating world of genetic engineering and CRISPR gene editing, exploring the scientific principles behind these technologies, their promising applications in various fields, the recent advances that are shaping the field, and the complex ethical and social dilemmas that accompany this powerful ability to edit. The Book of Life. Unraveling Genetic Engineering. Manipulating DNA. Genetic engineering is a field of biotechnology that involves the direct manipulation of an organism's genetic material. The goal is to alter the characteristics of that organism in a specific way, whether by adding, removing, or modifying genes. Traditional genetic engineering techniques, developed over decades, have enabled significant advances, such as the production of human insulin in genetically modified bacteria and the creation of transgenic plants with increased resistance to herbicides. These approaches often involved the random insertion of genetic material into the genome, which could lead to unintended effects. The polymerase chain reaction, PCR, revolutionized the ability to amplify specific DNA sequences, making the study and manipulation of genetic material much more efficient. Restriction enzymes, discovered earlier, act as molecular scissors that cut DNA at specific sites, allowing for the insertion of new DNA fragments. Vectors, such as bacterial plasmids or modified viruses, are used to carry the genetic material into cells. Despite its success, traditional gene editing techniques were often laborious, expensive, and imprecise, limiting their widespread use and application in complex organisms. The CRISPR Revolution. Gene editing with surgical precision. The development of CRISPR-Cas9 technology marked a revolution in genetic engineering, offering a much more efficient, precise, accessible, and versatile tool for gene editing. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. These DNA sequences were originally discovered in the immune system of bacteria, where they play a role in defense against invading viruses. The CRISPR-Cas9 system works as a DNA search and cut system. It consists of two main parts: a Cas9 enzyme, a protein that acts as molecular scissors, capable of cutting both strands of the DNA molecule at a specific location; a guide RNA, sgRNA, a small RNA molecule designed to be complementary to a specific DNA sequence in the genome that one wishes to edit. The guide RNA directs the Cas9 enzyme to the target site in the DNA. Once the guide RNA binds to the target DNA sequence, the Cas9 enzyme makes a precise cut in the DNA at that location. This cut triggers the cell's natural repair mechanisms, which can be exploited in two main ways: non-homologous end joining, NHEJ. A quick repair mechanism that often introduces small insertions or deletions of nucleotides at the cut site. This can lead to the inactivation of a gene. Homology-directed repair, HDR. If a DNA template with the desired sequence is provided to the cell along with the CRISPR-Cas9 system, the cell's repair system can use this template to correct or replace the DNA sequence at the cut site with high precision. The simplicity and versatility of the CRISPR-Cas9 system have made it an incredibly powerful and widely used tool in laboratories worldwide for a vast range of applications. The possibilities of gene editing, curing diseases and beyond. The ability to edit genes with precision offers enormous potential in various fields. Gene therapy for genetic diseases. CRISPR opens the possibility of correcting the genetic mutations that cause inherited diseases such as cystic fibrosis, sickle cell anemia, Huntington's disease, and many others. The approach can involve editing genes in somatic cells, body cells that are not sperm or egg cells, affecting only the treated individual, or, more controversially, in germline cells, sperm, eggs, or embryos, which could lead to heritable changes. Development of new therapies for cancer. CRISPR can be used to modify immune system cells so that they recognize and attack cancer cells more effectively, enhanced CAR-T cell therapy, or to inactivate genes that promote tumor growth. Creation of improved agricultural crops. CRISPR allows for the precise editing of genes in plants to increase yield, improve resistance to pests and diseases, increase tolerance to adverse environmental conditions, such as drought and salinity, and improve the nutritional value of food. Basic research and understanding of biology. CRISPR is an invaluable tool for studying the function of specific genes, allowing scientists to unravel the molecular mechanisms that govern biological processes. Development of animal models for diseases. CRISPR facilitates the creation of animal models with specific genetic mutations, which are essential for studying the progression of human diseases and testing new therapies. Eradication of vector-borne diseases. CRISPR is being explored to genetically modify disease-transmitting mosquitoes, such as those carrying malaria, dengue, and Zika, making them unable to transmit the pathogens. Recent advances. The frontier of gene editing. The field of CRISPR gene editing is constantly evolving, with rapid advancements that expand its capabilities and address its limitations. Base editing allows for the precise conversion of a single nitrogenous base, A to G, T to C, etc., in DNA without the need to cut both strands, which can reduce off-target effects and increase safety. Prime editing, an even more precise technique that allows for the insertion, deletion, or substitution of small segments of DNA at the target site with high specificity and fewer side effects. An improved delivery of the CRISPR system. Researchers are developing new and more efficient and safer ways to deliver the components of the CRISPR system, Cas9 enzyme and guide RNA, to the target cells and tissues in the organism. Regulation of gene expression. In addition to editing the DNA sequence, CRISPR is also being adapted to regulate the expression of genes, turning their activity on or off without altering their sequence. CRISPR in human therapy. Clinical trials using CRISPR to treat various genetic diseases and cancers are underway, with some promising initial results. Ethical debates. Navigating the implications of gene editing. The power to edit the DNA of living beings raises profound ethical and social debates that need to be carefully considered. Germline editing. The possibility of editing genes in sperm, eggs, or embryos, which would lead to heritable changes passed on to future generations, is particularly controversial. Concerns include the long-term safety of these modifications, the potential for unintended consequences, and the possibility of use for non-therapeutic purposes. Designer babies. Genetic enhancement. Editing genes beyond the treatment of disease, aiming to improve traits such as intelligence, physical strength, or appearance, raises questions about fairness, justice, and what it means to be human. Access and inequality. If gene editing therapies and technologies become available, there will be a risk of exacerbating social inequalities, with only the wealthiest having access to these technologies. Informed consent. In somatic gene therapy, ensuring informed consent from patients is crucial. Regarding germline editing, the consent of future generations is obviously impossible to obtain. Ecological consequences. The genetic modification of organisms can have unpredictable consequences for ecosystems, especially if genetically modified organisms are released into the environment. Safety and off-target effects. Although CRISPR is more precise than previous techniques, there is still a risk of edits occurring at unintended locations in the genome, off-target effects, which can have negative consequences for the organism. Playing God. For some, the idea of manipulating the code of life represents an ethical transgression, encroaching on a domain that should be reserved for nature or a divine force. Regulation and the future of gene editing. The need to regulate the research and application of genetic engineering and gene editing is widely recognized. Different countries and organizations are developing guidelines and regulations to ensure the safety, ethics, and responsibility of using these technologies. There is an ongoing debate about which applications should be permitted and under what conditions. Most scientific communities and regulatory bodies have adopted a cautious stance regarding germline editing in humans, with many advocating for a moratorium or strict restrictions. The future of gene editing will be shaped not only by scientific advances, but also by the ethical and regulatory decisions that society will make. Continuous dialogue between scientists, ethicists, policymakers, and the general public is essential to ensure that these powerful technologies are used responsibly and for the benefit of humanity. Conclusion: responsibly navigating the genetic future. Genetic engineering and CRISPR gene editing represent one of the most promising and challenging scientific advancements of our era. The potential to cure genetic diseases, improve agriculture, and expand our understanding of biology is immense. However, this power comes with great ethical responsibility. As we continue to unravel the secrets of the genome and refine the tools for its editing, it is crucial that we proceed with caution, guided by strong ethical principles and open and informed dialogue. The decisions we make today about how to use these technologies will have a profound impact on the future of human health, food security, and our relationship with the natural world. The ability to edit DNA is a frontier of knowledge that requires humility, wisdom, and an unwavering commitment to the well-being of all life. Responsibly navigating this genetic future is one of the greatest challenges and opportunities facing humanity in the 21st century.